this is an instrument that is used in a lot of drinking water utilities and a lot of research programs, and within governments in the US and actually worldwide, as a screening tool for algae monitoring programs. So really, the FlowCam originated from 3 marine biologists based at the Bangalore Laboratory for Marine Sciences in Maine. And it came about because they were frustrated because there wasn't an automated imaging counting system for marine phytoplankton. So they got together and they took the principles of flow cytometry and microscopy to make this flow imaging microscope. And they published their findings in Marine Ecology Progress Series in 1998. And they were able to report that they had successfully created this instrument, and they were able to take field samples and accurately image and count them. So why develop a FlowCam? You know, we've got microscopes, of course. You know, why spend all this money on this instrument? But, you know, anyone who's spent time on the microscope knows that it's time consuming, it's tedious, and you don't get results for, it can take hours really to run a sample. If you send your sample to a contract lab, sometimes you don't get the results back for about 2 weeks. So as we know, you know, things can change very rapidly in a lake or reservoir. So if you're waiting 2 weeks for your sample results, it's not really helping you. With the FlowCam, you can run a mill of sample in about 6 minutes. It takes a few minutes extra to classify your sample to determine exactly, you know, if you want to go down to the genus level. The data is stored on the instrument, so you can always go back and pull it up. You know, 6 years from now, if you want to go back and double check or train somebody on the data that you have from a really nice set, you can always pull that up. That's not something that's really possible with a microscope unless you take images of every cell that you see in there. So it's really a handy tool for A, getting rapid results, and B, you know, reproducibility. So this is the first FlowCam. You can see it's enormous. I think this is on a research vessel. And it's got a lot of different parts in there. This is the first prototype that was developed for sales. We called it the coffin. And this is the FlowCam today. So it's a really nice, sleek benchtop instrument. The computer is housed in the instrument, and then you're able to view your data on the attached screen. So it's basically a imaging system that takes multiple measurements to create statistically significant data. So your sample is going to pass through the flow cell. It's aspirated by a syringe pump. We have this high intensity LED and the fast shutter speed of the camera. So it's going to basically freeze your particles so that you're able to take this image. It can take thousands of images per second. And we also have the ability to add fluorescence to this. So we have these 2 photomultiplier tube detectors here, and they'll have filters so that we only capture the wavelengths that we're interested in. And so as the particle flows through the flow cell, the laser is directed to the center of the flow cell. And as it passes through the flow cell, through the laser, it will trigger a fluorescence event. So basically the camera will capture the image of the particle and it assigns a fluorescence value to that particle. And so from there, you know, we're able to determine what is a phycocyanin producing organism versus a chlorophyll only producing organism, and we're able to sort it based on that information. And so this is just a little bit more information about our laser. So we actually have 3 different lasers. We have the 488 nanometer laser, 532, and a 633. The 633 is the one that we term the cyano. So this is the one that we are using for HABs and is the most common in our drinking water plants here. And this is the one that has our 2 channels of emission. So the 700 for chlorophyll and the 650 for phycocyanin. And this is basically our home screen for the software. This is the flow cell, so you're able to see sort of in live view as the particles are captured, and they're being captured on the right-hand side here, and stored in the instrument along with all of the particle data. I think we have about 40 different particle properties that are measured. And so once you have your data, you're able to then sort them based on different morphological features such as size and shape and color. You can begin to semi-automate your analysis, so you can start building libraries, which you're able to then compare against your results, and this will speed up your analysis. And then we're also able to use the pigment to separate our results based on the chlorophyll and phycocyanin. And this is just an example of how that looks. So this sample was sorted based on channel 2 to channel 1 ratio. So we can see here we've got this number displayed below, and this is how we're separating what is a cyanobacteria versus other algae and diatoms. You can see, I have to keep moving my screen here. The number below this diatom here is significantly different from this cyanobacteria, and it enables us to sort our data. And then we're able to classify by functional groups. So the FlowCam will do this automatically. You run your sample and you can immediately see what you have as far as cyanobacteria, diatoms, and algae and detritus and decomposing. So it's very easy to, so yes, you're able to classify by these different groups, and then you can go further to the genus level if desired. But really, just looking at these three right here can give you a very good overview of whether or not you potentially have a problem in your lake or reservoir. And from here, you can export your data. It's going to give you your particles per mil for each classification, but it will also give you, you can choose to export with biovolume, area, a number of different particle properties. So it gives you a lot of information about what's going on in your lake. So I wanted to give you a couple of really great case studies. This one actually is from the National Institute of Oceanography in Goa, and they were kind enough to let me use this publication as a case study for today. And so this is looking at FlowCam measurements in 2 selected time series along the southwest coast of India, so they can explain how they respond, how the phytoplankton are responding to eutrophication associated with coastal upwellings. And so in addition to using the FlowCam for their phytoplankton measurements, both before and after the southwest monsoon, they're also looking at physical properties such as chlorophyll, temperature, salinity, and macronutrients such as nitrate, phosphate, and silicate. So this is kind of taking what Chris does and the FlowCam and looking at all of this data at once. And so the results showed that there were changes in size, morphology, in the living habitat of the phytoplankton. So the size increased during the monsoon period. There was an enlargement of cells and formation of much longer chain colonies, as you can see in these images here. And the change they found was due to adaptation of the microplankton communities to the turbulent conditions, allowing them for better uptake of nutrients. So this was their ecological response basically to nutrient enrichment. And so this is a great study. I know they're still working on a lot of studies out there in Goa and using their FlowCam for this. So we have a lot of FlowCams in research institutes looking at all kinds of eutrophication studies, but we also have them, as I mentioned earlier, in government agencies. And so this case study from Tennessee Tech University actually took 11 different partners to work on a HAB response plan for their state. So this is something that's, we're seeing it's handled differently with each state in the US. Some health departments are coming up with a sort of overall plan for the state, while other cities are kind of working on it individually. And in this case, they wanted to have one response plan for the state of Tennessee. And so Tennessee Tech University had a flow cam. What had been happening was that samples were being sent to a contract lab. And as I mentioned before, it was taking a long time for their results to be returned. By that point, there was really no HAB response to be done because it was too late. And so switching this up and sending the samples to Tennessee Tech allowed them to really rapidly identify and enumerate what types of cyanobacteria they had in the samples. These were coming from different habitats, so reservoirs, rivers, streams, and ponds. And once they had run it through the flow cam, they were also doing light microscopy because they wanted to see whether the types of algae, the types of cyanobacteria they had could potentially contain the gene for the toxin. And so this is kind of an example of one of their samples. You can see they ran their flow cam for the sample. They got their cyanobacteria, their detritus, and their other algae and diatoms. And it looks like about 90% of their sample were in fact cyanobacteria. So this triggered them to do further analysis through the microscope, where they were able to break it down to the species level and determine whether or not they needed to respond. Again, the flow cam can be used actually to go to the genus level. So this was their particular model, but a lot of different utilities are choosing not to use light microscopy because they don't really have the knowledge of how to speciate, and just being able to go to the genus level is sufficient for them to make those treatment decisions. So really, overall, it's a really great tool for rapid particle analysis. If it's used in the drinking water utility or in research, you can look at community composition and you can determine whether or not you have troublemakers. So you can look at trends over time and develop trigger levels for action. And using that fluorescence has enabled us to more accurately and rapidly determine what's going on, looking at your cyanobacteria versus your other algae and diatoms. So if you use the flow cam versus sending your samples out, you're really going to have much more rapid results. You're going to be able to act accordingly. And obviously, again, this is a monitoring solution. This isn't a treatment solution, but it can be used, as part of that whole overall solution to monitoring and treating your lake. So thank you.